We made it. Thanks for being here. We've made it to the final plenary on the last day, and this plenary is being hosted by ESEB, the European Society of Evolutionary Biology, for this joint meeting. Being Europeans, this morning will be relaxed and cool, and we can all have fun. I will introduce the main speaker. Oh, sorry, I should introduce myself. I'm Michael Ritchie from the University of St Andrews in Scotland, and I'm the president-elect of ESEB. Just before we get on to the main subject in hand, I wanted to see what the closed titles made of a Scottish accent. Are they working? Because the ones in St Andrews don't work for me, so I was curious to see if they worked here. Anyway, before we get to the main event, I would like to just introduce Tony Gabaldon for a couple of minutes. And Tony has the hard job of trying to persuade you that Barcelona is a cool place to visit.
Hello, everyone. So I'm Tony Gabaldon from the IRB and BSc in Barcelona, and I have the hard task of organizing the next ESEB meeting in Barcelona, and the easy task to convince you that Barcelona is a nice place to go in August next year. So you are all invited to come to the ESEB 2025 meeting that is going to happen in August at those days that you see there. And it's hosted by the Spanish Society of Evolutionary Biology and ESEB. And well, I guess most of you know Barcelona. It's a nice city, nice food, nice places to visit, Gaudi architecture, and many things to visit. So if you come here, please take some days extra to get to know the city. So the conference is going to be hosted in the International Convention Center of Barcelona, which is a place quite similar to this one, very modern with big halls and all the resources you need for a meeting. It's actually located in the Forum area of Barcelona, very close to the beach. And it's next to the Natural Sciences Museum, and all the delegates of ESEB meeting will have free access during that week to visit the museum, which at the time will have a special exhibition on species extinction, which I think will be of interest. And we have a very nice lineup of speakers. These are the keynote speakers that will open the session every day. And we are now with an open call for symposia, so you are welcome to submit your ideas for symposium. The call finishes in November, and then we will pick the symposia and we will open the call for abstracts. And in the first QR, you have a 5% discount if you pre-register to the conference. So you can do this now. It shows your interest that you are probably going to the conference, but it's not commitment at this point now. And with all that, I'd like to finish and give the floor to Mike again. Thank you.
Thanks, Tony. Now for the main event, I want to introduce Tracey Chapman to you. If you don't know Tracey, a quick potted history. She did her PhD in early research career in Edinburgh, mainly working in the lab of Linda Partridge, the very influential Linda Partridge, you might say. Then she moved to University College London in 1995 as a postdoc and obtained a Royal Society University Research Fellowship, before then moving to University of East Anglia in 2007, where she's been ever since and is now a professor of evolutionary genetics. Tracey has had a huge impact on the field, mainly because of her work on mechanisms of sexual selection and sexual conflict in Drosophila. I've been attending lots of symposia at this meeting, particularly the new ones on social fluids and the like. And I don't think it's an exaggeration to say that every other talk has been directly or indirectly inspired by some of the work Tracey did early on, which more or less established that field that's become so much broader and enriched now, even and spreading out to other organisms and becoming a universal topic that people are really trying to get to grips with, all inspired by her foundational and axiomatic work in that area. She doesn't just work on that, she also works more broadly on the genetics and biology of social interactions, some cool work recently in molecular drive mechanisms, aging and branching out into the applying some of these lessons to pest species. She's also a very active society member, not just in ESEB, but also for the Society for the Study of Evolution and other societies. I think she's served on 5 or 6 editorial boards, and most recently she finished a 4-year term as editor-in-chief of the journal Evolution, and has also been the vice president of the Society for the Study of Evolution. She's done lots of work for ESEB, been on council, been on policy committees, been on international committees, and obviously at the moment is now our president. I just finish with a very quick personal comment, if I may. There's a bit of overlap between our CVs because as an undergraduate, I was in Linda Partridge's lab shortly before Tracey went there, and then I did my PhD at University of East Anglia before Tracey went there. And I remember particularly back in Linda's lab, you know, I was very young and I was full of sort of imposter syndrome, like what am I doing trying to be a scientist? And the thought that several decades later, Tracey and I would both be here doing this sort of thing is amazing. So please welcome Tracey Chapman.
Thank you very much indeed for that overly generous introduction, Mike. Really appreciate it. Okay, thank you for coming. What a tough gig this is. So it's the final morning, it's 8 o'clock for Christ's sake, and you're here. So let's hope we've all got enough caffeine on board to get us through this. So I really appreciate you coming along. I also would like to thank the members of ESEB for electing me as president. It really is a true honor to serve in this position. So Mike has introduced me, but as he said, I am currently based at the University of East Anglia, which is about 2 hours sort of northeast of London on the train. This is a spectacular part of the world in an understated way. Giant skies, really rich natural history, archaeology, paleontology. It's one of the few places in Europe where there's evidence for occupation by 4 different hominin species. So come along, please come and visit. If you're a student of architecture, it's also an amazing place to go to. So this is at the bottom there. This is the kind of world-famous neo-brutalist concrete. We have a lot of award-winning concrete in my university. And the whole university where I work is very young. It was designed in the early 60s by Dennis Lasden, an associate and very famous architect. And he designed the whole outfit, the whole layout of this university along a sort of long linear structure called a teaching wall, whose explicit aim was to break down barriers and to have a non-hierarchical organization of a university where academics and support staff and anybody would necessarily have to bump into each other and talk to each other, hopefully. So I think that's really an ideal that I can sign up to, even if it's often difficult to achieve in practice. Okay, so along the way, I have been fortunate enough to receive advice and mentorship from many influential and really important people that were important to me, and I want to acknowledge that debt and the support and advice that was given to me by these people. These gave advice freely, often very freely, if you know some of the characters involved. And I didn't always listen to it, but I really value their advice and support. Okay, so I'm going to talk this morning about sexual selection, So one of Darwin's big ideas, so the form of selection which is focused on traits that ensure success in reproduction. But before I get on to that, I want to take this opportunity to say a few things about ESEB, the importance of ESEB and of scientific societies in general, and also some of the really pressing challenges that we are facing. And we need your support, we need your help in addressing these challenges. And so ESEB, has a number of aims, activities. We support the study of organic evolution. We promote this scientific view in research and education. We publish journals. Our flagship journals are the Journal of Evolutionary Biology, Evolution Letters, which we co-publish with SSE. We organize congresses, as we just heard. ESEB 2025, open for business, pre-registration is open. Running through, a core theme of all of this activity is the desire to be inclusive. So inclusive to our membership and who publishes in our journals. So this kind of activity is important. We're interested in it, and therefore it's important. And it's important because we're interested in it. And that's a kind of sort of circular argument, but I think it's always worth taking a step back and thinking, well, what is the absolute importance of what we study? What's the absolute importance of evolutionary biology? And I think, of course, studying the rules of life. So the origin of the first replicators and everything that follows from that, how mutations occur, how they're accumulated, selection, everything. And I can't actually think of a bigger question than this. Maybe if I'm pushed, the origin of the universe might be a bigger one. And so this is incredibly important to us, and we should recognize that there's an underpinning importance of the subject that we study, regardless, whoever you are. And also I think that scientific societies have a great role to play in valuing knowledge and the nature of knowledge and protecting this. And actually what I mean by that is that, for me, there's a kind of non-negotiable way of understanding what we think is important, why some ideas are important and why others are not. There's an objectivity to that regardless, and I think that's really important, protecting and nurturing the nature of knowledge. And scientific societies as well have a great deal to do in terms of ensuring that our processes, our data is rigorous, we protect and we ensure strong ethics and so forth. So I think these are just really important, always worth remembering why we're doing this. Okay, but there are some challenges, some real challenges for scientific societies. So we are not nearly as inclusive as we need to be. We know that. We need to do more. Our journals there is a huge number more journals in the area of evolutionary biology, evolutionary ecology than there was 10, 15 years ago. Those that are faster, that are sort of shinier, perhaps, than our society journals might be perceived. And so how do we maintain their competitiveness? How do we maintain their reputation and their relevance? Are our society journals publishing the right sort of thing, or are we all sending our best, what we think of our best stuff in a particular area somewhere else? And meetings as well. So hopefully, by the time we're here on the fourth day, we've been able to see some of the benefits of being at a meeting like this. But there is an environmental impact of meetings like this, which we have to consider. So I'll talk a little bit about that. So how do we address these challenges? What can we do? I'm going to talk through these things. So we have, along with other societies, we have a bunch of measures to try to increase inclusivity. And we have an Equal Opportunities initiative, details here. We also have a Global Evolutionary Biology initiative. Here this establishes local researcher networks, teaching curricula. We support our members, of course, members to come to the meetings. So particularly members from low-income countries, to, sponsorship to come here. The Godfrey Hewitt Mobility Award. Godfrey was a wonderful colleague who I only got to know in the latter part of his life, and he was such a champion of ESEB and supporting fieldwork and lab visits and allowing people to take their own new research direction. So this is something which we also sponsor and think is important. So that's all important. I think we can all sign up to that. That's great. Facilitates a lot of stuff, but it costs money. Where do scientific societies get their money from? So we get money from membership, somewhat, although we try to keep costs low, and we get money from journals. But journals, it's a very, very competitive nature out there, and as you will probably know, the nature of academic publishing is really changing very, very rapidly, and who knows where we'll be in 5-10 years' time, but this is what we have now, and it needs our support. We need to submit our high-quality research to our society journals. Okay, so it's Tuesday morning, it's truth time. I want you to raise your hand if, when you are thinking about where to send a paper, your primary consideration is whether it's a society journal or not. Can you just raise your hand? All right. Actually, I can't see anything at all, so I don't know. I don't know. But I hope that I think at least my friends in the front row had their hands up. And but this is what we want you to consider. We want you. It's not saying you have to, but have this as a primary consideration of thinking about where to publish your research. And if we send our best quality research to our society journals, or consider doing that, or perhaps consider coming to these, once we've gone somewhere else and been disappointed, as often happens, then we can increase the competitiveness and the reputation of our journals. Also, I think it's important to recognize the quality and the value of scientific society papers. It's hard to get into these journals. We're a really picky bunch, I think. We rigorously review each other's papers, and we are really, concerned that the thing is really well supported. And so, some might say we're sort of overly picky, but it has such a fundamental importance, this rigorousness, that is evident in the work of the society journals. And we need to recognize that. If we are going to do more than pay lip service to, the scientific declarations, DORA, which are trying to encourage us to move away from journal-based metrics and to look at the individual quality, then, this is something which we can do when we're evaluating work and applications and so forth, to recognize the value. And of course, to participate. So review for scientific journals. Of course, I'm focusing here on the ESEB journals, but I also really have in mind, AMNAT, Evolution as well. All right, participate, review for us, and get involved. Become an editor if you have the opportunity. What we can also do to support our memberships is to create and celebrate success. And so I want us all to give a big shout out here. So the Distinguished Fellow Award is our highest honor in ESEB. And if you were lucky enough to hear the 2023-24 winner, Ophelia Rantz, gave a fantastic talk in the virtual symposium of this meeting in June. So I want to give a great round of applause, please, for Ophelia. It's fantastic. Celebrating success, creating award winners here. So yesterday afternoon, it was a fantastic symposium we had of our JMS prize winners, and this is a prize for outstanding early career researchers. And we also had talks from the EUEA Awards, that's very difficult to say, and so the Underrepresented ECR Achievement Awards. And this was fantastic, the standard and quality was so high. And those of you who were there will know, actually, it was also incredibly moving to hear the stories and the histories of people from who were talking from the EUEA, just stop. That award. And so I want you to please give a huge round of applause to Magdalena, Emily, Olivia, Paul, Zabibu, Sophia, and Harihar. Fantastic achievement. Thank you. Okay, something which ESEB does, which I really like, is to try to initiate opportunity as well. So to try to make research and science happen that doesn't otherwise happen. And we fund a bunch of special topic networks. And again, if you were at the virtual meeting, you'll have seen that there was a symposia with presentations from these. So these are networks. We fund, give funding so scientists can come together, have meetings, and hopefully initiate and synergize some research that wouldn't otherwise happen. And so this has the advantage of trying to help us to remain current by stimulating research in areas that we think is important. And many of these networks, they come together to develop shared protocols or shared databases. For example, picking one in particular, not just because it's run or partially run by a colleague of mine, and I'll probably get into trouble if I don't pick this one. But this is a evolutionary ecology of thermal fertility limits. So a group of people who've come together and are doing really fantastic work on systematic maps and systematic meta-analyses of the impact of rising temperatures on fertility, actually showing that populations are being limited by their fertility limits and not their viability limits. So, we're very proud of this kind of research that we could do, and we want to do more. Okay, meetings. So what's the challenge of meetings? So, we can reduce the environmental impact of meetings by encouraging, the reduced use of single-use plastics and putting more plant-based foods into Congress offerings, but it's plane travel. Plane travel is responsible for more than 90% of the impact of Congresses. And in the face of the existential threat of climate warming, we need to act. And so, led by Craig Primmer and Anya Westrom, ESEB 2027 is going to try an experiment. We're scientists, we like experiments, so we will try. And we have resolved to run ESEB 2027 in a dispersed format, and there will be 4 hubs within Europe. And the idea is to reduce the need to fly. Okay, people within Europe, will hopefully attend a hub which is within a train travels ride. Of course, people, not everybody can do that, and of course, people will fly. And it's important to say there are no restrictions. There's no formal restrictions on which hub you go to or how you get there. But the aim is to reduce the overall impact in terms of the number of flights. Or, if you fly in maybe, to Europe, perhaps you don't fly again. So they reduce those short-haul flights as well. The hubs will be interconnected in a way. I think it's quite hard to visualize how this will work. So this is a little schematic, but you've got global plenary. So you've got plenary speakers, which will be in each hub, but for each plenary. Actually, this should be keynote, not plenary. Nobody knows what the word plenary means, but anyway, sorry, I'm sort of a bit of a pedant. Okay, so there will be keynotes connected, and there will also be parallel sessions. So the same session which is streamed across the hubs, but there will also be a local flavor. So each hub will have the same local sessions, but that session will have a local flavor. And the idea is that, attending any one of these will be an equivalent experience for the people involved. And of course, if we do that, we have to evaluate it. If we do the experiment, how will we know whether we've succeeded? And so we've had to form a whole new committee to evaluate and to sort out how to evaluate this, to establish a baseline, to ask people about their experiences, and to see whether we've actually achieved our aim of reducing the environmental impact. If it works well enough, and we've got ways to improve, we'll intermittently in the future repeat this experiment. And of course, it's important to say, as for anybody who's considering, whether a meeting like this should intermittently be, an entirely virtual meeting, this will not be the same. This kind of meeting cannot be the same as a meeting like this. We have to just accept that. But, it will be different, and, it will be valuable, we hope. So we need your help. If anything that I've said was of any interest to you, want to get involved, please contact us. Okay, so I want to move on to science now. So here I want to talk about the evolution of reproductive traits, and so traits that serve in competition between individuals of the same sex or serve as the basis of female choice displayed by individuals of one sex and chosen by the other. So I want to talk about the drivers of that and also some really interesting and exciting work, I think, on the kinds of reproductive traits that are plastic. But sexual selection, of course, isn't just about, you know, this extraordinary elaborated traits that we see here. It has a genome-wide significance because measures of quality or success of sort of higher quality individuals leave an imprint across the genome. And, you know, which has the, well, theory predicts, and there's empirical evidence that this can purge deleterious mutations, can protect against extinction. So, you know, this big, one of these big ideas of Darwin is important, doing important and global stuff. But it's the form of, another big idea that arose in the 1970s or so is sexual conflict. So within sexual selection, interactions between the sexes that are that can be conflictual is an idea that emerged in the 1970s, although of course there were antecedents of this in the population geneticists of the 1930s or so. If I forget to say that every time, Brian Charlesworth will find out about it and have another go at me. So, you know, Brian, done it. Okay, but it was really Jeff Parker who formalized this idea, which we're going to evaluate, by empirical studies on these yellow dung flies, which was formalized into, you know, theory. And the idea is that there is selection on male mating strategies despite the costs that are exacted upon females. What is the idea? So we're talking about an interlocus sexual conflict, so a scenario where the fitness interests of males and females are distinct. And here we're talking about diploid outbreeders, where, you know, individuals that are unrelated are coming together to reproduce, and there will likely be multiple mating episodes with different partners. And so if we think about a trait, there are lots of reproductive traits that are sort of emergent property of the interaction of two sexes. So, you know, how many offspring to produce at any particular reproductive bout, or how frequently to mate. But the value of that trait may have different optima in males and females. Perhaps females might gain in any particular reproductive bout by producing slightly fewer offspring and males slightly more. So we've got the value of the trait is shown by the vertical line here. And so there are different optima. So the value of that trait can't simultaneously, you know, be optimized for males and females. So that's the idea. And so fitness interests of males and females in a scenario like this, it's a true type of genomic conflict. They can't always be aligned. And this can drive adaptation and counter-adaptation as each sex is selected to shift the value of a trait like this towards their own optimum. And it has the potential to drive even rapid evolutionary change. The sorts of factors which will cause this to become intensified, I guess, is how far apart are the optima to start with. What's the current value of the trait? What's the relative power that each sex has to do anything about this? Is there even any mechanism that can be deployed by either sex to affect the value of this trait? And so what I want to do is just describe some of the work that we've done to sort of evaluate this idea using the fruit fly system. And so we work primarily for this strand of our research with Drosophila melanogaster, which even amongst people who are into, you know, insects is really the kind of persona non grata, insecta non grata, if that's even a thing, right? But you know, we use it because it does, you know, it does have a really incredibly rich behavioral repertoire. And the tools, the genomic and genetic tools that are available are very rich and really allow us to perturb systems in a way that helps us to understand what are the forces that you know, are governing the evolution of a system like this. And then hopefully we can, from there, identify targets to go and look in natural populations or make things, you know, more realistic and find out, you know, what's the relevance in a more ecological context. Okay, so we've done a lot of work on the role of a particular really charismatic group of proteins and peptides, the seminal fluid proteins of male Drosophila that are transferred along with sperm during mating. And the hypothesis that we've been testing is what's their role in mediating the balance between conflict and cooperation. Now, of course, the sexes have to cooperate in order to ensure that reproduction occurs effectively, and a whole series of events that have to happens in the right order, and it is likely that these molecules help to do that. But there's also the potential for conflict, as I'll say. So there are more than 200 of these. They're mostly made in the accessory glands, which is, if you think about it, the sort of insect equivalent of the mammalian prostate. And they're hugely diverse. There's more than 200, probably more even than 300. The number's just going up all the time. And this is why they are interesting. They have this huge variety of really, fundamental effects on reproduction. So there are seminal fluid proteins that can modify female attractiveness, female sexual receptivity, how often the females sleep, how much they eat, what they eat, how many eggs they lay, how many eggs they ovulate, how sperm get into storage, get out of storage, are used. This is a sort of really fundamental global kind of reprogramming also of almost of female behavior and physiology. And of course, not surprisingly, this has an important impact on the fitness of both sexes. And there's a huge amount of work by a huge number of different labs that have contributed to this body of work that I'm summarizing here. So why do we think there's the potential that these molecules can potentially mediate interlocus conflict? And so, well, if we reduce seminal fluid protein transfer by males, so if we ablate genetically the accessory glands, costs of mating by females, they are eliminated or significantly reduced. Seminal fluid protein transfer can also benefit males while simultaneously exacting costs in females. And also, if you have a sort of multi-mating context, a female mating with males in series, and in the standard sort of wild-type situation, everybody there has a different interest in, what the interval between the different matings should be in terms of what would maximize their overall reproductive success. But if you make the female unable to respond to a key seminal fluid protein, you find that those interests converge. All right, and so also the existence of, costs in females, so as a result of this sort of battling between males and females, one would expect that females that are exposed to high levels of conflictual interactions should evolve counter responses, which indeed they do. Several studies show that, and I'll show you an example later. Okay, but there's been how important is this? Maybe you push systems and you can see that this is a factor, selective factor, which can shape. What's usually the function? What's the sort of standard? How important is it? Is what I'm trying to say. What's the balance between conflict and cooperation? And there's been quite a recent discussion of this in the literature, which I welcome, because it's, of course, there is going to be a balance. But the important and difficult thing to work out is how important is the kind of conflict that I've described, in a sort of natural situation. And of course, this list of things that I put up here is not just specific to the difficulty of finding out what selective forces are involved just in this system. It's much bigger than that, right? And so what kind of phase are you in the system? Are you in the origin of a particular kind of conflict, or the spread, or its maintenance, or its resolution? Perhaps conflict resolution is a big study, and there's evidence that the genome is full of sort of evidence of past evolutionary conflicts which have now been evolved. But, that's quite a hard thing to do. So sort of looking for something which isn't there anymore and which, can have some challenges. Okay, and there's a lot of context dependence as well. And I'll give you two examples. And so in a way, the field has sort of arrived at a kind of, it depends sort of area, which is interesting. But fitness measures are key. If we don't really understand the ultimate consequences of the changes or the manipulations that we're doing, then it's very difficult to infer. And so one example of a sort of context dependence is shown here. And so these are event history diagrams, if you know them. And so each row here is an individual female. Okay, so it shows you the lifespan, how long a particular female lived, and the colors for each row show you how much reproduction that female had. So high in red and going down to low in yellow. And so you've got two groups here, and this is a free-running experiment. We're not controlling anything here. We're not trying to partition out, effects of mating or how often seminal fluid proteins are transferred. We've just got a free-running situation where females are kept with males a lot, high male exposure or low male exposure. And you can see, they're not the same. They're very different. There's a huge survival cost for high exposure to males. The reproductive patterns are sort of slightly different. So, if you're trying to work out here what's best, right? You can't do that without considering fitness and probably age-specific fitness. And in fact, it depends, right? This is individual fitness expressed as a function of the population growth rate. So either of those kinds of outcomes, could either be costly or not costly to females, depending upon population growth rate. Another example from female condition. This is an experiment which is designed to test whether the condition, so the nutritional condition of individuals, affects their ability to respond to potential conflict. And I think here a concept that we've borrowed from the social insect, social evolution literature is really useful. So you can have the potential for a conflict, it will be there, inevitably because of the principles that I outlined earlier, but whether it's actually expressed will depend upon a number of different factors, one of which is condition. And so this kind of experiment using experimental evolution can address that idea. And so this is a cartoon of the sorts of regimes that you can have running over many, many tens of generations. You can keep individuals in female-biased, FB, equal sex, or male-biased regimes. So you have this isn't the actual experiment. They're bigger than this, and there are replicated lines. And of course, the prediction would be that females from the male-biased regimes will be subject to elevated levels of sexual selection and sexual conflict. And so if you're going to see the evolution of any resistance against aspects of that exposure that are harmful, you would see it there. And so if you take then females from those regimes after running them for many, many tens of generations, and you expose those individuals in a common garden to standard types of males, shown in green, and then you look at the survival, what you'd be expecting to see, if there is indeed the evolution of resistance against those elevated levels of sexual conflict and costs, you expect to see this, which is what you see. So females from the male-biased regimes are living longer in the presence of males than are those from the equal sex or the female-biased regimes. Okay. But that's what happens if you are providing ad libitum feeding, and if you run alongside a regime like that, individuals that are protein and carbohydrate restricted, you don't see that pattern. So it's not because that potential conflict isn't there, because the same intensity of selection is being exacted. The females are not able to cannot express resistance. So that's a context dependence, which I think is quite important. Okay, so that's really all I wanted to say about drivers. I'm just trying to throw a few ideas out here as well to see what you think. In the last part of my talk, I want to tell you about a new line of research that we've been doing on reproductive plasticity, because I think we have been surprised by the extent to which the sophistication with which individuals are plastic to their social and sexual environment, and the fitness effects that this has. And so I think perhaps we shouldn't be surprised, everything is kind of cleverer than we think. But also I was going to say that this whole strand of research really came from observations, just noticing things in the lab. And I was reminded of a presidential talk that Butch Brodie gave for the ASN, where that was a message that he was saying, you look at things, think about what you're seeing. And so I really like that sort of serendipitous aspect. So the context is that sexual interactions occur within a context. You've got groups of varying numbers of males and females present. And so individuals that have the potential to respond in an appropriate way to those conditions have the potential to gain fitness benefits. Now, where I'm going with this is the bottom line, this is what I'm going to say, is the nature and the consequences of plastic responses is really different in males and females, and the fitness outcomes. And so this is just in case, you want to tune out for a bit. It's the last day. So this is where I'm going. This is the take-home message. So if you need a few minutes to yourself, take it. Okay, so first off, the plastic responses of males. So we investigated, what are the effects of males on other males? Can males potentially respond to the threat of sexual competition and act in a way which increases their fitness? And so quite remarkably, the length of time that you expose a male to another male, even before they meet a female, will determine the length of time that they will then mate with the female for when they meet one. That was slightly garbled. Anyway, you can see there's quite a strong relationship here. And so the longer that males, the more males are perceiving the potential threat for competition, they are plastically increasing the duration of mating when they meet a female. Now, so what? Okay. Well, there are significant fitness benefits of those extended matings. So females mated to males that were previously exposed to a rival will take longer to remate. So that's the purple bar over the white bar here. Those matings have increased fecundity. Those males have higher success in sperm competition. So there are fitness benefits for males. They respond to rivals. They mate for longer. What's actually going on is this. You show a male another male, and within 2 hours, some really remarkable changes to gene expression happen. So they switch on all of these sensory genes in the top part of the animal, the head and the sort of thorax. And then in the reproductive system, with the abdomen, the accessory glands, they're switching on all of these ejaculate protein genes. They're making more ejaculate, just following exposure of males. And, I'm going to show you an example here. So for one key seminal fluid protein, sex peptide, I'm going to show you the amount of that peptide that's actually transferred into females by males that were previously exposed to rivals or that weren't. And in each case here, so the red and the blue lines, it doesn't matter for the purposes of this what those are, but in each case, when the male was previously exposed to a rival, he's putting more of this peptide into females. So the squares are higher than the circles in each case. Okay, so males responding to males, switching on these ejaculate genes, mating for longer, transferring more to females, getting fitness benefits. What we've also been surprised by is that, there is a lot of genetic variation in the extent even of that plasticity. And this is just a snapshot of the many tens of lines that we've tested. And so these are individuals drawn from the DGRP. So this is a reference panel developed by Trudy McKay. And what you've got here is a bunch of inbred lines extracted from a natural population. So if you imagine the wild-type natural population, all that genetic variation is partitioned into different inbred lines. And so then if you examine your trait of interest in each of those inbred lines and you get significant differences among the lines, you can take that as evidence that there is some form of genetic variation for the trait of interest. And so what we were doing here, something rather complicated, but we were trying to assess what's the degree of plasticity, so the degree of difference between the grey bars and the white bars for each of these lines. And sometimes you get lines that respond really, really strongly to the presence of rivals and some that don't. So there's genetic variation in the extent of plasticity. There's also genetic variation in the amount underlying that, the amount of these peptides which actually put into females. So this is really interesting. You know, what's maintaining this? I mean, there are some, you know, costs of course, but you know, if it's better to be a male that transfers more only when they need to, which I guess is the point. You know, that's the significance of plasticity. Why aren't they all doing it? And one idea that we're testing, and I'll be interested in your views on this, is that there are multiple routes for males to be successful. And so here are some fitness surfaces. So fitness against two key components of males' reproductive success. So how competitive they are in sperm competition, and what's the interval before the female that they've mated with mates again. And so you can see for the control sort of wild type plot at the top, you know, there are different ways for males to be successful. There's a peak in one place, but then there's a why is there a valley? Why is there a valley there? That's not, I don't know. And then there's another peak at the front. So, you know, you can either be, you know, a very good sperm competitor and rubbish at preventing your female from mating again, or you can be good at both. And when you reduce the amount of seminal fluid proteins in the system, that's the bottom panel, you get fitness peaks in all sorts of different areas. You know, and there's going to be a whole range of variation of, as I've shown you, how much of this stuff is put into females. So, you know, this is an idea that we're evaluating and trying to work out whether we've actually got, you know, sexual specialists, or generalists here, borrowing some from some theory from the ecology. Okay. So that's male. So the plastic responses in females, so just a few minutes on this, is all about, is very different. They're not responding to other females in the same way at all. Their plasticity is all about how many eggs to lay and where to put them. And so females show very dramatic plastic fecundity responses to the social and the microbial environment. I don't know if you can see, but in the top dish, there's a bunch of eggs dusted all over the top, and then in the bottom, there's a bunch of eggs that are clustered. And this is a plastic response that all females can make. And some of the things that shape it are the social group size, and that's the panel in the middle, and also whether there are likely to be microbes present. So just to elaborate and add a little bit more, you know, in order to, well, we think the clustering looks kind of non-random, but is it really non-random? And so we've done some modeling and then simulations and fitting to the actual data. You know, within a given space, how many eggs could be put there, and where are they actually put? And so the result of that simulation and fitting parameterized by the data is shown here. You have a clustering preference which increases with the number of females that are present. And that's kind of interesting because an observation that we were very surprised by is that those clusters are often of mixed maternity, and you can show that if you dye the eggs in various different ways. And so there's a bunch of white eggs from one female and pink eggs from another here. And that's not what should be happening. If you put all your eggs in one place together with those of other females, you're increasing direct competition. So this is a really interesting phenomenon to try to understand what's going on here, which allows us to test actually some really general theory. You know, perhaps there's some public goods benefits here. Sometimes eggs have antimicrobial or anti-cannibalistic substances on them in some species. Maybe they're diffusing out. You know, maybe we've got free riders. It's an interesting evolutionary problem. Perhaps there's a selfish herd. Maybe the individual probability of predation for eggs is reduced if they're in a cluster. Perhaps there's some other sort of abiotic protection. So that's very much the kind of line that we're going on at the moment. Finally, so I said, you know, it's all about fitness. So I just want to tell you some work that we've done to try to work out the broader fitness consequences of this, you know, mishmash of who's with who, to look at the fitness consequences of this plasticity. And so first I'm going to show you some work that we've done to look at the actuarial patterns of aging for individuals, females and males that are exposed to same-sex and opposite-sex environments. And so what I mean by actuarial aging is, you know, lifespan is rather a sort of, you know, poor measure of how you get there. You know, you can look at maximum lifespan, but is that after a lifetime of poor health, or is it, you know, very good health and then a sharp decline? And so actual actuarial aging, is about, when aging starts. And so that's on the x-axis here. And then once it starts, how rapidly does it proceed? And that's shown on the y-axis. And so we've done experiments with huge numbers of fruit flies in boxes, and replicates of all of those, for females and males that are exposed to these different types of regimes, the treatment. So, individuals will reproduce once, and then, they have a lot of same-sex interactions, or they can reproduce intermittently, that's in pink, continuously, that's in red, or you can jack up the density a bit and increase the same-sex interactions. And so when you do that, for females, this is what you get. So I've just put some arrows on to help us understand this complicated plot. Basically, if you start from the gray circle, as you increase reproduction, you increase your aging starts earlier and occurs more rapidly. But if you increase same-sex interactions, you get something which goes in an opposite or contrasting direction. So for females, this is females I'm showing you here, which is a bit of a surprise. Bit of a surprise that females kept with other females have this dramatic effect on one aspect of aging at all. But the interesting part, when you superimpose males in there, okay, so that's the plot. It's in opposing directions, completely opposite. I mean, there's less going on. They are less sensitive overall to these social and sexual environments, but what differences there are in an opposite direction. So that's really, the fundamental structure of actuarial aging is completely different in males and females upon these different types of exposure. And so that's actuarial aging. What about fitness? And we've now done an experiment to look at that, equalizing density. And I'm not going to say a great deal about this because Lauren Harrison gave a really excellent talk about this yesterday. So if you want to know more, you should talk to her. But if you look at the fitness effects for individuals that are exposed to same-sex, opposite-sex, mixed-sex environments in each sex, you find that there is strong reproductive senescence in females and not males, and sex-specific effects of these same-sex interactions, which are surprising. Okay. So they matter. Fitness consequences of plasticity are doing important things. The fundamental basis is different in males and females, which perhaps fits into this conflict paradigm which I've been describing today. The nature and consequences of plastic responses are different in males and females. I'm repeating this in case anybody's been asleep. It's all right. You haven't missed anything. That's the conclusion. Okay, so that's really it. That's all I wanted to say. So hopefully, an illustration of some of these concepts with our insect Drosophila systems has been of interest, and I'd be really intrigued to know your thoughts. So it just remains for me to thank my current team there on the left. The people in blue here are actually at the meeting, and the people whose work I've particularly focused on have an asterisk. And so, obviously really like to thank all of those people who just make everything possible, do all the work, and the funding bodies. And also, I'll just finish by thanking you again very much for attending this morning. That's it.
Time for questions. You want to field questions yourself?
Sure, yeah.
We've got microphones at the sides if anyone wants to ask questions. It's actually hard to see from up here.
We did it. Anyway, we finished before 9.
Yeah, well done.
It's on. I think he said it's on now.
Hello.
Hey.
Oops. Hi. Thank you, Tracy. My question is related to this thing of the eggs clustering together.
Yeah.
Did you, I'm a bit ashamed to say that I have never seen Drosophila eggs. So did you try to see whether there are fitness consequences of that? So like putting them apart versus having them together, or having them with kin versus non-kin,
Yeah.
does that change their fitness, like
Yeah,
survival or something.
Yeah, so on the kin angle, Tristan Long has done some really nice work on this about cooperative feeding as a consequence of where the eggs are laid in relation to kin and non-kin. And so I would defer to him. So there is some work on that. But what the puzzle for us is we've tried to do exactly what you've you know, very intelligently proposed, and are finding it difficult to find out what it is. And I just, you know, I could have shown you some of the experiments that we have conducted to eliminate most of the things that I've suggested. But, you know, one particular thing is testing the selfish herd. We had an experiment where we were looking at predation by ants. So ants will take Drosophila eggs and will pull them out. And so we think, okay, if there's a cluster, they can pick off the outside ones and perhaps leave the ones in the middle. And actually what they do is they come along and they pick up the whole blooming cluster, you know. It's brilliant. It's like the total opposite of what they should have done. But, you know, it's a really important point. And, you know, that's actively what we're doing at the moment. So hopefully, maybe, you know, but next year by Barcelona, we'll know what the answer is to that. Thanks for the question.
Oh, I'm just going to hold. I'm curious about the plastic response of males to rivals. It's hard for me to imagine in nature that there are males out there that aren't regularly encountering other males. So do you think that plastic response is actually selected in nature, or could it be a lab adaptive effect or something else happening?
Yeah, it's really, that's a really good question. And, you know, of course, that's an important one for all of this work, isn't it? You know, to what extent are the principles that we can identify here in controlled conditions relevant? All I can say is that there is indication that, you know, because the amount of the length of exposure that males to other males and the initiation of the gene expression changes that I showed, that's entirely consistent with what happens when you have aggregates of fruit flies in a natural context. So that's not at all the same as showing that it's relevant. But, you know, as far as we are aware, the conditions exist for these things to occur. But thank you very much.
Hi, thanks very much. I actually had a question about your earlier remarks about society journals. The science was fabulous, but I just had a quick question, and that is, you alluded to this briefly, the challenge of you know, the need to submit to society journals. One of the big pressures, I think, is tenure and promotion committees. And you alluded to this briefly, but a lot of the reason why tenure track faculty submit to, you know, what are perceived to be the top journals is because of tenure and promotion committees, which are often composed of non-scientists who use these metrics such as nature to, you know, clue them in as to what's important or not. So I'm just curious what you'd recommend about getting around this issue, because that is a major pressure, I think, to steering people away from society journals.
No, thank you. That's a really important issue. And of course, I, you know, sidestepped that. And I actually, you know, didn't put up my hand when I asked that question. I mean, earlier. I mean, I do think about that now, but I, you know, didn't used to, for precisely, you know, the reason that you said. So it's really, it's difficult, and I get it. And one would hope that, and I don't know if people are aware of this, you know, DORA convention, which is trying to get away from journal metrics, but we talk about it, but we don't really own it, I think, because what everybody does when they look at faculty promotion stuff is they go to the journals and they look, you know, and as you said, non-specialists will look at what they perceive is the reputation of the journal, and if you don't know a journal, you go and look at those metrics. So I don't know. I don't know what the answer to that is. I mean, I think we need a kind of culture change and to kind of bring up the reputation of our society journals, so that they are held to be, you know, more important than they you know, than perhaps they currently are, or to recognise, you know, the value, like, you know, 3 really good papers in Evolution Letters or AmNat, you know, is as good as something, you know, something in a kind of shinier, flashier journal. I mean, that's it. I want, you know, I'd be interested to know what you think. I mean, certainly there's a re-education job that needs to be done, and, you know, our university across the UK, across Europe, and I don't know, you know, quite how much it is in the States, is really doing a, you know, an education job to the people who are assessing promotion cases and reviewing faculty job applications to try and get away from that. But there's still a huge amount of work to do. I don't want it to be a council of despair, though. Anyway, thank you. I don't know who was next.
Good morning. That was a beautiful talk. I have one question about the biology and one comment about the journal discussion. I guess I'll start with my comment. So at my university, which is in the US, we have a very powerful and wonderful faculty union. And through the union, we have very transparent guidelines for tenure. So luckily for us, we don't have those kind of, I mean, they exist, but they're more transparent. So it is easier for us to choose a society journal over a Charnier for profit. So that's one thing, just one comment there to add. My question about the biology is about the actuarial analysis, which is fascinating stuff. Are there, and you may have mentioned this, I apologize. Are there different phenotypes, I guess, or different responses before and after first mating? Like if the female is mated and then the analysis is done, and then prior to that first mating, because there is huge, phenotypic changes before and after that first mating event.
Yeah. So thank you. Thank you for the question and the comment. So it's the actuarial analysis that I presented. These boxes are kind of free running. So we don't know who's just. We don't know those things. Certainly, if you look at the patterns of aging in females that haven't mated versus those that have, you get different patterns. So there are different responses. And I don't know, I guess, it's your point that needs to be kind of factored in, or it would be good to partition them out, or?
I guess that would be my suggestion. There is definitely work in the immune world. The immune responses of females before and after first mating are remarkably different. There is some beautiful biology there to be explored.
Yeah, no, I agree. And, there is, as many of you probably know, there's a huge biogerontological industry which works with Drosophila as a model system to partition out those things. I guess here we were trying to get at, okay, in this situation, we talk about it and we've got this kind of control thing with a few individuals and tubes. What if we bung them all in a box, and what happens? Because I think it's worth emphasizing that, those experiments have to be on a huge scale, as you will know, because otherwise you cannot with certainty detect reliably those actuarial estimates. And so, but the compromise, you do it on absolutely massive populations, is that you don't know much about the individuals involved. Thanks.
So actually I wanted to come back to the idea of journals and evaluation, and I wanted to support your claim then and say that a lot of our institutions or universities have signed the DORA declaration. And so often they sign them, but maybe they don't really use. So when we are sitting in panels and committees, we have to remind that our institution have signed that and make it to be really, impactful and have an effect on the way we're doing evaluation. So I know that you can do that by sitting in a committee and just, recalling that we have signed that and we should stick to it. Another thing that, so I was in France sitting in the CNRS evaluation committee for a while, and there's now some databases like Daphne. I don't know if you heard about that. That is actually kind of ranking journals and giving like journals that are okay or not okay, and society journals or not. And we are increasingly using this, and people are using that in their curriculum, in their application to talk about their ethics of publication. So I think we can change that. Even an evaluation is really the crux, but we have to be really, really pushing for it. In particular, when we are in the position of evaluating those careers and promotions.
Yeah, no, I agree totally. And of course, I I do get it. And going back to the comment earlier, of course, in some countries, the impact factor of a journal is the explicit metric which is used, and promotions also depend upon publishing in journals at certain, so it's hugely important. And there's a lot of work to be done. So it's really good to hear about these, initiatives that are happening. But it's sort of arrogant to stand here and say, like, oh yes, you need to do this. And that's not what we're saying at all. In a way, we have to change the culture to make our journals, better, and to, as I was saying, to get everyone to recognise the value of the research that's in there, because it will have been rigorously, reviewed. And more importantly, because that's right. That's the right thing to do. But also, it supports all of the wonderful and enriching activities that all the societies have been talking about here, and that are under threat if we don't support our journals.
A perfect presidential address. Fantastic science and lots of really important stuff about the future of our societies and academic publishing. Join me in thanking Tracey again for her talk this morning.
Thanks.